Dear friends, this video is about trisomics in crop improvement. There are variation in chromosome number as well as structure. The trisomic condition comes under variation in chromosome number. There are two main types of variation in the number. One is euploidy and one is aneuploidy. Euploidy means numerical change in the entire genome, whereas aneuploidy is change in the number of a one or few chromosomes. In euploidy, there are different conditions like monoploidy, that means one set of genome represented as X. Haploidy, only the haploid or gametic set of genome represented as N. Diploidy is two set of genomes represented as 2X. Polyploidy is more than two sets of genome. In that, triploid, tetraploid, pentaploid, hexaploid, etc. conditions can be observed. Aneuploid condition is further divided into hypoploids and hyperploids. Hypoploids has monosomy, double monosomy, nullisomy, double nullisomy like conditions. Monosomy is loss of one chromosome from the diploid set, that is represented as 2n-1. Double monosomy is 2n minus 1, that is loss of one chromosome from diploid set from two different chromosome sets. Nullisomy is loss of one chromosome pair from the set, that is represented as 2n-2. The second condition is hyperploids, that means additional presence of chromosomes along with the diploid set. Trisomy is addition of one chromosome to set 2n+1, and tetrasomy is addition of a pair of chromosomes, that is 2n+2. That means hyperploid has trisomy and tetrasomy conditions. And now in this video we are going to discuss about trisomy, that comes under the numerical chromosomal aberrations under aneuploidy and the subdivision hyperploids. Okay. This is how the structure of chromosomes are represented in different hypoploids and hyperploid conditions. In trisomics, you can see the three chromosomes are located at one place instead of normal two. This is also a representation of trisomy condition. Aneuploid due to gain of one extra chromosome is nothing but trisomic condition. The primary trisomics means trisomics is further divided into three types: primary, secondary and tertiary trisomics. Primary trisomic is normal chromosome, secondary is isochromosome, and tertiary is translocated chromosome. There is further division like telotrisomic or telosomic trisomics, acrosomic trisomics, fragmented trisomic, double telotrisomic, double isotrisomic, double tertiary trisomics, isotertiary trisomics, isotelotrisomics, and telotertiary trisomic. In the last section, the sixth classification whatever is given, that comes under compensating telotrisomics. The primary trisomics, as you can see, the normal chromosome we can find means extra chromosome is normal. In secondary trisomics, the extra chromosome is isochromosome, that means both arms has same chromosomal arm. In tertiary trisomic, it is a translocated one. In telotrisomic or telosomic trisomic means only one arm is present, one is absent. Acrocentric trisomic means the telomere at one arm is absent and the centromere is at acrocentric position. Fragmented trisomic means the both arms are missing or both telomeres are absent. Compensating trisomics means one arm is compensating for the other, like double trisomic means on the one chromosome is normal and rest of the two are having compensating type, like one chromosome is having left arm and another as right arm. Double isotrisomic means isochromosome is present, and in the double tertiary trisomic means here you can find the translocation kind. Likewise, the classification goes on. Now the question arises, why trisomics are used in diploids? Normally, aneuploids used for cytogenetic studies for diploids are different from those used in polyploids. Hypoploids means X monosomics cannot be used in diploids, as diploids cannot tolerate the loss of a chromosome. However, they can easily survive in polyploids. Thus, monosomics used in polyploids and trisomics can be used in diploids. And these are the two conditions used in plant breeding as compared to other chromosomal aberrations. Origin and source of trisomics. As I have already discussed, that there are three things: primary, secondary and tertiary trisomic. Coming to primary trisomics, here unequal disjunction means non-congregation and non-disjunction that is found in Datura. Asynopsis and desynopsis condition found in maize, cotton, wheat, tobacco. Triploidy condition can be observed in tomato and rice. Autotetraploidy in maize, potato, haploids in wheat, translocation heterozygotes and tetrasomics in Datura. These all are the source of primary trisomics, and by the way how the primary trisomics occur. This is a photograph showing non-disjunction. In the meiosis 1, when non-disjunction happens, the two pictures represent, one is normal and another is abnormal or non-disjunction occurred cell at the meiosis 1. And when non-disjunction occur at meiosis 1, what will happen at the meiosis 2? There will be unequal transmission of chromosomes to the progeny. So after the meiosis 2 division, or when the gametic formations are observed, what will happen? There we can find the trisomics and monosomics in equal proportion. In the second case, you can find where the non-disjunction occur at meiosis 2 condition. Here the proportion of obtaining gametes with different chromosomes is 1 with trisomics and 2 with diploid condition and 1 with monosomic condition. Coming to production of primary trisomics from triploids. When triploids are observed, what will happen? When gametic condition are observed, it may give N and 2N condition, and in the further segregation it may give N plus 1, N plus 2, N minus 1, N minus 2. And this the triploid condition that will segregate into N plus 1 and N plus 1 plus 1 further crossed to normal disomic condition. So when F1 are observed, it will yield trisomy and double trisomy condition. That is, remember, when triploids are crossed with normal disomics, that will yield trisomy and double trisomy. That may give other kind of progeny also, but for our clarification here I have taken trisomy and double trisomy. Coming to methods of production of secondary trisomics. How the secondary trisomics are observed: by monosomics in wheat, primary trisomics in datura, tomato, barley, maize, asynopsis and desynopsis in case of pea, triploids in pea, monotelodisomics in tomato, monoisodisomics in tomato. Origin of iso and telocentric chromosomes. When misdivision happens, the left arm and the right one, they will misdivide, and that division will lead to the integration of left and the different chromosomes into one. That will give rise to isochromosomes, or two telocentric, one isochromosome, and four telocentric chromosomes. Production of tertiary trisomics from primary trisomics. When primary trisomics are crossed to translocation homozygote, that will be having condition like 2n plus 1 and normal translocation homozygote with normal 2n condition, and that will give rise to tertiary trisomic, that is 2n plus 1. Tertiary means one arm is translocated. Identification of trisomics. By their morphology, like extra chromosome has significant effect on morphology and physiology. Each trisomic has distinct morphology. Extra chromosome affects development of many parts of plants. Example, primary trisomics in tomato. Triplo 1 means extremely stunted, weak, poor growth in field. Triplo 3 and 4 means susceptible to blossom and drought. Triplo 1 to 9 means perform better in greenhouse than in field condition. Triplo 5 is smaller and compact growth than diploids. Triplo 8 means glossy convex surface on leaves at early stage. This is how the different stages are represented, and the observations are made in IR 36 rise. Identification. First one we have seen identification based on morphology. Now identification based on karyotype. Trisomics can be identified based on chromosome morphology based on banding pattern, Paquetin studies, banding techniques like C and N, or it can be seen by fish or Gish techniques. Coming to different configurations of trisomics in meiosis. The primary trisomics form trivalents during meiosis. Chain configuration is more common than fry pan and Y-shaped configuration. The type of configuration formed depends on Paquetin pairing and number and position of chiasmata. Long chromosome means some arm can be involved in chiasmata, that is fry pan is common. Small chromosome means chain trivalent is common. Here different shapes are given. One is chain, fry pan, Y-shaped and triple arc. Secondary trisomics. In this, chromosome can form trivalent, bivalent plus univalent and triunivalent. Ring trivalents are most common in secondary trisomics. The different shape or configurations are given in the picture, like Y-shape, fry pan shape, stick shape and all. Different pentavalent configurations in tertiary trisomics. Exclusively the tertiary trisomics form pentavalent form, and that is most common. Transmission of extra chromosome from trisomics plants. Two types of gametes are formed in trisomics. One is diploid, that is X plus 1 condition, then tetraploids, 2x, 2x plus 1, hexaploid, 3x, 3x plus 1. These are formed when trisomics are normally present in the plants. In many cases on female side, gametes with the extra chromosome will be functional without any competition. In male side, there is competition between X plus 1 gametes for transmission. In diploid gametes with X plus 1, failed fertilization cause no germination, poor germination and slow growth of pollen tube, swollen tip of pollen tube and slow rate of growth of pollen tube. Coming to the applications of trisomic. In plant breeding, these are used for assigning linkage groups to specific chromosomes, mapping of genes on specific chromosome arms, locating the centromere and orientation of linkage groups in hybrid seed production. Trisomics can also be used in disease or biotic and abiotic stress resistant studies or inducing the condition. Genetic ratio expected from trisomic segregation. Genetic ratio of factor located on trisomic chromosomes will be different from the ratio of genes on disomic chromosome. Genetic segregation ratio of trisomic individuals are a means of locating genes on chromosomes. This is how locating recessive gene is observed. When 7 trisomic stalks are taken in barley, the trisomic stalk may be 1, 2, 3, 4, 5, 6, 7 chromosomes are taken here, and that is crossed to disomic plant. Here trisomics is female and trisomic is male. 7 F1s are trisomic plants, and among 7 F1s we have to identify the trisomic plants, and that trisomic stalk on chromosome, for example, is trisomic is observed on chromosome number 2, and that is further crossed to disomic plant with recessive condition. In the F1 we can get the, that is called as non-critical cross, we will get the heterozygous condition, and by selfing that what will get 3:1 of normal diploid segregation, and here gene is not present on chromosome number 2. That is how we can locate the recessive gene. Critical cross in trisomic stalk for chromosome number 1 into disomic, what will get, the gametes are homozygous capital A, or we can say the dominant AA, and one more is single A, and the another gamete is small a. After crossing this, in F1 we will get capital A, small a, and capital A, small a, that is in heterozygous condition. There in the F1 we should identify the trisomics and it should be selfed. The gametes what we obtain is given here. Here n+1 gametes not transmit through males. The phenotypic ratio observed is 17:1, significantly deviate from the 3:1 normal disomic segregation. Gene is present in chromosome number 1 here. Coming to mapping of genes on specific chromosome arm. For this again the trisomic is crossed to normal recessive disomic condition. In the F1 we should identify the trisomics and we should self. After selfing, all the trisomic progenies expressed dominant traits, that is all dominant and no recessive. Diploid progenies of selfed trisomics show 3:1 segregation. Non-critical cross of selfed progenies segregate in 3:1 ratio. Assigning genes to specific chromosome arm. Here there is a big table is given here for assigning specific gene to particular chromosome. How we can do, we can see where in the F2 progeny and secondary trisomic or telos trisomic or tertiary trisomic, if we want to get all in dominant form and none in recessive form, we can easily identify with the help of different cross combinations or by backcross progeny or F2 progeny when we obtain secondary trisomics, telotrisomics and tertiary trisomics after a particular cross. Here the things are given in red form. The same process or trisomics are used in construction of the chromosome maps of rice. Coming to balanced tertiary trisomics for using in hybrid seed production. The process was given by Ramaje in 1960. Characteristics of balanced tertiary trisomic. In this, extra chromosome is interchanged. Nuclear gene for male sterility is located close to the breakpoint. Marker genes for identification may be red plant color or plant height, etc. Prerequisites for balanced tertiary trisomics are: male sterile should not be pollen transmitted, BTT should be vigorous and competitive, and the major thing is it should be adaptive or having great stability or adapted to different agroclimatic situations. Stable male sterility across environment. This is the flow chart how the BTT is done. We'll just cross the trisomics, tertiary trisomics and normal diploid one. In BTT, two fields will be grown separately. In one, the field is exclusively kept for seed multiplication, or we can say the field for producing continuous male sterile plants, and one more is for the production of hybrid seeds. In the first case, when the tertiary trisomics are crossed to normal disomics, and when the seed maintaining of the seed is done, in that condition, in the F1 we'll get 30% BTT plants. That 40% is further divided into two groups, 15% and 15%. 15% will further go for the maintenance of seed to make the male sterile plants, and the 15% will be contributed for production of hybrid seeds. Like that, the technique is mainly to enable the male sterile material or the A line continuously for the production of hybrid seeds and throughout the year. Here 70% diploid female and 30% of BTT are obtained, and 70% diploid female are discarded, and 30 will be further utilized for further process. This is how it is done. Here BTT production field is different and hybrid production block is different. In hybrid seed production, 95 to 100% hybrid seeds can be obtained. Thank you. I hope you liked my video, and do not forget to subscribe.